So today we are going to talk about male reproductive system, and it's very important to understand how it is organized, even in case you are not going to be andrologist, because in any way you have to understand how different drugs you prescribe affect the process of spermatogenesis and male reproductive function. You know that male infertility now is the relevant problem of the 21st century, and to understand how to cure it, what factors do affect the reproductive function, we need to understand how this function is organized under normal circumstances. So let's start to investigate directly from the seminiferous ducts. Seminiferous ducts, this is the system of tubules where spermatozoa are formed and through which they are transported out of the male reproductive system. So the first site where spermatozoa appear, these are the ductally seminiferi contorti. Inside the testis, you know, there are nearly 250 lobules, and in every lobule there is a loop of convoluted seminiferous duct, or ductally seminiferi contorti. And inside inside these tubules, spermatozoa are formed, and further they are transported to the tubuli seminiferi recti. Here they are straight tubules. Then rete testis. Rete means network, you know, and it is located in the mediastinum testis. And out of here, spermatozoa are transported through the efferent ductules, or the ductuli efferentes, to the epididymis, because ductuli efferentes, they represent the head of the epididymis. Then through the ductus epididymis, they are transported to the ductus deferens. Here it is in this diagram, ductus deferens, and then through the ductus ejaculatorius to the urethra. So this sequence you have to know, and now we are going to discuss them in details. First of all, we have to highlight that spermatogenesis takes place in the testes, and they are put in the scrotum outside the body cavity. Why they are put why they are put there? Because for proper spermatogenesis, the temperature should be a little bit lower than inside the body. And to decrease the temperature, they are put outside, and at the same time they have a special arrangement of blood vessels. So they have their artery passing through the rich network of veins. So veins, they contain blood, cool blood, returning from the scrotum, while arteries, they bring warm blood from body cavities. So warm blood passes through the network of venous blood. In this way, it decreases, it cools down a little, and it helps to maintain the stable temperature inside the testis. So, you know, if to increase the temperature around testis, if they are not descended due to some defects of embryonic development, then spermatogenesis is violated, of course. So let's start discussing the seminiferous tubules directly from the ductuli seminiferi contorti. Here we can see a scanning electron micrograph of the frozen specimen, and we can see that on the cross section there are tails of spermatozoa. So here they are formed, at the same time they are fixed to its walls. And if we make the cross section, we'll find out there are two layers. The layer of the seminiferous epithelium, so this epithelium generates spermatozoa, and also there is a layer, of tunica propria. It is made up of several layers of myoid cells, as well as fibroblast-like cells. So those cells are needed for peristaltic movement, to transport spermatozoa out of the testis, as of course spermatozoa are immotile in this stage of development. And now we are going to consider in details the seminiferous epithelium, as this is the site of spermatozoa formation, and this epithelium is represented by two types of cells. These are the Sertoli cells. You may see here giant supporting cells assisting in spermatogenesis, and spermatogenic cells. Here they are of blue color on different stages of their development. So spermatogenic cells, they start to appear at the base of the seminiferous epithelium, and ascending upwards, they become more and more mature, eventually detaching from the surface and transport and further moved along the seminiferous duct system. So let's consider those spermatogenic cells in details, along with the process of spermatogenesis. First, we have to say that spermatogenesis starts at puberty. So before the puberty, spermatozoa are absent in the male organism, and the process is guided predominantly by the testosterone, and it continues along the life of the male individual. In spermatogenesis, we consider 4 phases. I want to highlight again, in different textbooks, you have different description of spermatogenesis. That's why please pay attention exactly to our slides and to our lecture, because we ask you and we are awaiting to hear from you exactly this answer. So during the first phase, we call this phase reproduction. So spermatogonia, precursors of spermatozoa, spermatogonia, they divide mitotically, increasing in number just by mitosis. After that, they undergo the process of growth, and you may see that you can see here the cells are the largest, and we call them primary spermatocytes, and they grow, become enlarged, and that's how they differ from spermatogonia. Spermatogonia is smaller, spermatocytes are larger. Despite both are formed due to mitosis, they both have diploid number of chromosomes like all the other somatic cells. Then, process of maturation involves 2 consequent meiotic divisions. So maturation here means that their nuclei become mature to perform their function. But despite 2 meiotic divisions, despite their nuclei are formed and mature to perform the function, their morphology is not fit to perform the function. So during the last stage, spermiogenesis, spermatids, they do not undergo any divisions, but instead spermiogenesis is completely about morphological changes. So cells, they become, they acquire outer appearance of the spermatozoa. So all about the superficial changes. Notice, please, that this is the subject of confusion. Spermiogenesis is not the same as spermatogenesis. Spermatogenesis, we call this process in general 4 phases, while spermiogenesis is the only last phase. Okay, and it includes only morphological changes in the appearance. Okay, let's investigate spermatogenesis in details. So the first phase, reproduction, it involves mitotic division of spermatogonia. Spermatogonia are the first cells in the seminiferous epithelium, and they are located at the base, most close to the basal lamina, and they are the most mature. There are several subtypes of those cells, type A and type B. Respectively, type A transforms into type B, but among the type A we consider dark cells, reserved stem cells, and type A pale cells. So pale cells, they are going to transform into type B, so they are destined for further differentiation. Then spermatogonia, by mitotic division, divided mitotically, give rise to primary spermatocytes. And as I told you, primary spermatocytes, they enlarge in the size. And this diagram given here actually is not correct, because primary spermatocytes here are shown smaller than spermatogonia. That's not right. And not always diagrams correctly reflect the process. Okay, but you have to remember that primary spermatocytes, by the arrangement of chromosomes, are the same as in spermatogonia. After that, the secondary spermatocytes are formed, and here from primary to secondary, there is the first meiotic division. So secondary spermatocytes are formed through the first meiosis. As for the first meiosis, you have to remember, first of all, that there is a crossing over in its prophase. So chromosomes, they exchange their regions one with another, and that's why, as a result, we'll have chromosomes with paternal and maternal genes mixed one with another. So as a result, spermatozoa, they'll contain another combination of signs comparing with the maternal-paternal cell. So we have one, you remember, paternal chromosome, one maternal chromosome. As a result of crossing over, they exchange their regions. So after the crossing over, there is no completely paternal and completely maternal, but instead we can see two chromosomes, mosaic structure like that. So it is positive for the diversity. Yeah, it increases the diversity of the newly formed sperm. The second thing you have to keep in mind when we are talking about the primary meiosis, the first meiosis, is that as a result, chromosomes, they go to opposite poles. So you remember that we have 23 pairs of chromosomes, and in mitosis, every chromosome is split in the region of kinetochore, and chromatids are moved to the opposite poles. But in case of spermatogenesis, in case of meiosis, in the first meiotic division, one paternal chromosome and one maternal chromosome, they go to opposite poles. So we can't see here that chromosomes are split into chromatids. But of course, this is not paternal and maternal, this is the mixture, as we have said. But in the first meiosis, chromosomes, they move to opposite poles, not chromatids, but chromosomes. And remember that every chromosome contains two chromatids, absolutely identical one with another. Then, after the secondary spermatocytes, we have spermatids. And here in this process, they undergo the second meiosis. And in the second meiosis, now chromosomes are separated into chromatids, and now chromatids are moved to opposite poles. And what we have to keep in mind about the secondary meiosis, second meiosis, is that there is no synthetic period. So the number of chromosomes is not increased. The number of chromosomes is stable. So let's summarize it. From primary to secondary spermatocytes, we can see the reduction in the chromosomal number if we had 46 chromosomes with 2 chromatids in each. In secondary spermatocytes, we have 23 chromosomes, 2 chromatids in each. Then secondary spermatocytes, they give rise to spermatids. In spermatids, we have 23 chromosomes with only 1 chromatid in each. So the minimal number of chromosomes, that's why the nuclei of spermatids and spermatozoa, nuclei of spermatozoa are the smallest in the human body. Okay, so this is in general about meiosis, for you to recollect some knowledge from the first year. And now we have to discuss the last phase of spermatogenesis. This is the spermiogenesis. It is, in turn, is divided into 4 phases: Golgi, cap, acrosomal phase, and maturation phase. They all are about the outer appearance and morphological changes, as I told you. As at the beginning of the Golgi phase, we can see just a rounded cell without tail, without acrosome. At the end, we can see the mature spermatozoa with all organelles properly arranged. You remember that it has its head, neck, and tail. So what happens in the Golgi phase? Golgi creates a system of vesicles, and then in the cap phase they fuse with the formation of acrosome-like structure. And what is especially important in the cap phase is that nucleus get rid of its pores And inside the nucleus, all chromosomes, they become tightly packed. As you remember, instead of histone proteins, those chromosomes of spermatozoa, they use protamines. Protamines, they help to pack DNA more tightly than histones, because spermatozoa, they are not using their DNA before the fertilization. Yeah, so the nucleus is granted to the oocyte, and before granting it, it is not used. So all DNA is tightly packed, is completely in the heterochromatin. Then in the acrosome phase, the exonemal complex also is formed, and in the maturation phase, exceeds of cytoplasm are shed off, so the volume and the weight of spermatozoa is minimized because it should move for a long distance. So all the exceeds, all the excess of cytoplasm is lost and phagocytosed by the supporting Sertoli cells. Okay, so what we have to say about spermatogenesis in general, what is the clinical significance, and what you should know for your practice, medical practice, clinical practice. First of all, that in general it takes 74 days for spermatogenesis, from spermatogonia to the mature spermatozoa. What it means? It means if you want to affect spermatogenesis, for example, positively, if you want to stimulate spermatogenesis, then the result of those drugs or those treatment action could be analyzed only after 74 days, because exactly this period is needed to create spermatozoa. And opposite situation is true. If patient takes some drugs like antibiotics or another drugs negatively affecting spermatogenesis, then you should warn the patient about the reproductive function, that spermatogenesis would be affected, so it is better not to plan children, yeah, in the nearest future, in the nearest 74 days. So remember this term, it's very important. Another question arising from this term is that why spermatozoa are not produced once in 74 days, yeah? We know that spermatozoa are continuously produced along the reproductive period. And the answer is for the question how the spermatogenesis is continuous and how it is maintained continuously, is in that different regions of seminiferous tubules, we have different phases of spermatogenesis. We can see different phases. So if we make a cross section through the testis, we'll find out in every particular segment different stage. So spermatogenesis is not synchronized in the whole testis and in both testes. So in every particular region, different stage could be seen. So every particular moment we can see in some regions of seminiferous tubules, mature spermatozoa are formed. In some other regions, only the first stage, spermatogonia, only give rise to spermatids. And because of that, because of so-called waves of spermatogenesis, the process of spermatogenesis is continuous and is stable. And more or less stable number of spermatozoa are produced per one period of time. Despite the whole process takes 74 days, okay, because it is not synchronized. Okay, then, as we have just considered different types of spermatogenic cells, spermatogonia, primary spermatocytes, secondary spermatocytes, spermatids, and at last spermatozoa, we also have to discuss another component of the spermatogenic seminiferous epithelium, and these are the Sertoli cells. Sertoli cells, as you can see, they harbor spermatogenic cells and support them mechanically, so they assist in movement of those spermatogonia from the base to the lumen of the seminiferous epithelium, to the apex of the epithelium, to the lumen. And on its way, they support spermatogenesis by producing different biologically active substances and phagocytosing exceeds of cytoplasm and doing many other functions we are going to consider right now. Besides, what I forgot to say is that spermatogenesis doesn't break down all the connections between spermatogenic cells, and they continue to contact with intercytoplasmic bridges up to the last stage of spermatogenesis. Okay, so let's talk about the functions of Sertoli cells. First, of course, they provide mechanical and nutritional support because seminiferous epithelium devoid of blood vessels. So if we return here, we will see that spermatozoa are far from the source of nutrients. So deliverance, nutrients transport takes place through the cytoplasm of Sertoli cells. So mechanical and nutritional support. After that, they produce different substances, and you should know the effect of every particular substance. First, this is the androgen binding protein. What does it do? It binds testosterone and helps to increase locally its concentration. Then inhibin. Inhibin, this is the substance negatively affecting the synthesis of the follicular stimulating hormone. Later on at the end of the lecture, we are going to discuss that Sertoli cells, they are homologs of follicular cells in the female reproductive system. So Sertoli cells are activated by follicular stimulating hormone. And please remember, it is very important. So as they are stimulated by follicular stimulating hormone, by this, they produce inhibin to inform pituitary to stop producing follicular stimulating hormone. So inhibin, this is the kind of the negative feedback loop from the Sertoli cells. So inhibin is needed to prevent excess of follicular stimulating hormone production. Then Mullerian inhibiting factor. This factor inhibits female reproductive system formation. It inhibits Mullerian duct development. Okay, so it is especially important in embryonic development. Then transferrin, ceruloplasmin, these are the transport form to help transport iron and copper respectively. So please remember those substances and their effects. It's very important. Then, together with Mullerian inhibiting factor and SRY gene, they orchestrate the development of the male reproductive system. So by producing those substances, Sertoli cells participate in the embryonic development and they regulate the development by male line. Then phagocytose defective spermatogenic cells and also the phagocytose remnants of exudes of cytoplasm, and they provide blood-testis barrier. So as for the blood-testis barrier, we have to stop and to discuss it in details because it is very important. First, the reason of its existence. First of all, blood-testis barrier, it insulates genetically foreign spermatozoa from the bloodstream and from human immunity. So what it is written, it isolates genetically different antigenic haploid germ cells from the immune system of adult male. And it helps to maintain the high testosterone concentration. So by producing here those tight junctions, by exposing tight junctions, Sertoli cells, they help to concentrate testosterone locally. But in other words, what you have to comprehend is that spermatozoa, as I told you, they appear only in puberty. So before the puberty, our immune system is unaware about the spermatozoa existence. And in males, when spermatozoa appear nearly in the puberty, it could be considered by immune system as foreign cells because spermatogenic antigens are not familiar to the immune system, so they could be rejected. That's why spermatozoa are hidden from the immune system by this barrier. Yeah, and another reason is that spermatozoa, due to the crossing over, they exchange the different regions of their chromosomes, so they have a little bit another combination of genes. So this is another factor making them different from the paternal cells. So in any way they should be hidden. Then why this barrier is very important? Because if it is damaged, if barrier disappears, mechanically damaged, for example, then immune system starts to destroy spermatogenic cells. That's why even in case of traumatic injury of only one testis, another testis will be also rejected. So this is a very dangerous and it should be taken into account during the surgical intervention. So during the surgical interventions, seminiferous tubules should not be cut, and blood should not be exposed to the seminiferous epithelium, because in case it happens, then as a result, both testes will be destroyed by its own immune system. Okay, so what is the nature of this barrier? Actually, it is made up of tight junctions between Sertoli cells. Also in the formation of the barrier, actin cytoskeleton and smooth endoplasmic reticulum of Sertoli cells, they are also involved in the formation of those junctional complexes. But the most important component, of course, is the tight junctions. So tight junctions, they separate the seminiferous epithelium into two compartments, the basal, here it is, and adluminal, here it is. And in the basal compartment, only spermatogonia and early-spermatids could be found. But all the others appearing after the puberty, they move, they break down those tight junctions and they move upwards. And they exist in the adluminal compartment, which is devoid of blood vessels and which is insulated from the immunity. Okay. So after we have discussed Sertoli cells with their multiple functions in assisting spermatogenesis, in creating a special microenvironment, in producing factors and creating the barrier, we have to discuss another important population of cells. These are the Leydig cells or interstitial cells. They are called interstitial because they are located not inside seminiferous tubules, but in between them. So in the interstitial tissue, it is important. And here they are. What it is written? They produce, you should remember, testosterone, male sex hormone, one of the most important androgen. Then insulin-like protein 3. What is the function? First, you should remember, it controls the descendance of testes to the scrotum during the embryonic period. And what is the clinical significance? This factor is produced by Leydig cells, so by measuring it in the bloodstream, one can determine the steroidogenic capacity of Leydig cells. So by the presence of this protein, we can conclude the capacity, the activity of the Leydig cells in general. And oxytocin, you know, in females it stimulates the contraction of uterus, yeah, and here it stimulates the contraction of myoid cells surrounding the ductal seminiferi contortia, or the convoluted tubules. So those myoid cells, by contraction, they stimulate spermatozoa movement along the system of ducts. Then Leydig cells, they have a typical appearance of steroid-producing cells. If you remember adrenal gland, we also discussed their two organelles very well developed. These are the smooth endoplasmic reticulum and mitochondria. And the same is true for this place. Endoplasmic reticulum smooth and mitochondria with tubular vesicular crests. You remember that crests usually they are lamellar, like flattened, like lamellae. But in this case, tubules are formed. And this is the sign of steroid-producing cells. The feature of Leydig cells, these are the crystals of Reinke. So just remember, if you see anywhere in the MCQs or in any description of the cell, you should suspect Leydig cells. And then they are located in the space between the tubules, so do not forget their location, because the cells are very important. It is difficult to overwhelm their function. So you should know where to find them, what is the typical appearance, and of course, what's the function. Let's move on. After the convoluted tubules, reproductive testis is located, lined with a simple cuboidal epithelium, and reproductive is located in the mediastinum testis and is communicated with the afferent ductals. It is important to say that afferent ductals and intratesticular seminiferous tubules, they derive from different embryonic sources. So retrotestis communicates intratesticular ducts with ductally afferentes. Okay, besides here the specimen of the testis is shown. A testis is represented by convoluted tubules, and as we have already said, you may see in every tubule different stage of spermatogenesis. Here spermatozoa are absent, but here we can see tails of mature spermatozoa are ready to detach and to be transported along the duct system. That's why always continuously some amount of spermatozoa are formed, millions per day. So this continuous production is due to non-synchronized spermatogenesis. And here in the specimen, how you can distinguish different cells one from another. First of all, at the base you can distinguish spermatogonia, here they are. After that you may see the largest cells, and you know that the largest are the primary spermatocytes. Then a little bit smaller nuclei, these are the secondary spermatocytes, and eventually spermatids and spermatozoa. Then with the green arrow, Leydig cells are shown, and as you can see, they are located between the seminiferous tubules. Then, so as we have completed with the testis is a very important organ, with spermatogenesis. There we have discussed the spermatogenic cells, Sertoli cells, Leydig cells, and from the retrotestis we move to the Epididymis, and here we have to discuss that Epididymis is the organ in which spermatozoa are incubated for nearly 12 days. So after 74 days of spermatogenesis, nearly 12 days are needed to retain spermatozoa in the Epididymis to become fully mature, and it should be taken into account. So Epididymis is represented by the head, body, and the tail. In the head, there are afferent ductules. Here the head of Epididymis is. While body and tail, they are represented by the ductus epididymis, so it should not be confused. Ductus epididymis is present only in the body and the tail, and the structure of the head differs significantly. So let's investigate it. What's the difference between the afferent ductules and the ductus epididymis? In the afferent ductules, they derive besides from the mesonephros. We'll talk at the end of the lecture. In the afferent ductules, there are two types of cells. There are cells with microvilli and cells with a cilia. So different superficial structures are present. And also they are surrounded by the smooth muscle cells, as here spermatozoa are immotile, and in any way, even in case they are motile, they should be removed by peristaltic movement of the smooth muscle cells, and those cells appear only close to the ductus epididymis. Notice, because those two types of cells in the epithelial lining, they have different height, the epithelium is it has a different wavy shaped line, superficial line. That's because cells are irregular in the height. Okay, so the general function of afferent tubules is to reabsorb the fluid. Okay, now as for the ductus epididymis, this is a single duct, but very long, up to 7 meters, and it is highly coiled, so it is used as the storage of the sperm for some period of time, of course. And it is lined with a pseudostratified columnar epithelium, because there are some basal cells, of course, for regeneration, and columnar cells with the stereocilia at the top. And stereocilia, this is the subject of confusion, because despite it is called cilia, it is originally a microvilli. These are the long microvilli. So please pay attention and do not confuse, because it was thought this is the cilia, but after electron transmission electron microscopy investigation, it was understood that really this is the microvilli, but the name was preserved as the stereocilia. The name was not changed. Okay, so the smooth muscle coat, yeah, it increases its thickness from the beginning till the end, because in the ductus deferens, in the vas deferens, smooth muscle cells, they are arranged in 3 layers. So along those 7 meters, we can see continuous increases in the thickness of smooth muscle layer. And what is the function of the epididymis? The first function is decapacitation. You know that capacitation is when spermatozoa are incubated in the female reproductive tract to become capable of fertilization. So capacitation, when sperm become capable to fertilize. But decapacitation is opposite process, is when spermatozoa are covered with some molecules preventing preliminary acrosomal reaction. So spermatozoa are prepared for a long journey in the female reproductive system, and their surfaces become modified, and the process called decapacitation. Then in the epididymis, spermatozoa, they become motile only here. Yeah, the reabsorption of fluid takes place, and also different components are produced to affect the spermatozoa maturation, so creating the proper microenvironment. But if to be honest, sialic acid, glycoproteins, and modification of glycocalyx, these are the part of decapacitation process. So the last is almost the same as the first. Okay, so let's look at the specimen and try to distinguish, and let's reveal how to distinguish afferent tubules from the ductus epididymis. So here we can see the head of the epididymis with the afferent ductals. You may see the epithelial surface is of irregular height. That's because cells with the cilia intermingle with the cells with microvilli. Okay, so ductuli afferentes, or the afferent ductals. This is the head of epididymis. And here you may see that the surface of epithelium is smooth, cells are tall columnar, the epithelium is pseudostratified, and you may see perfect stereocilia at the top. Here they are very, very long microvilli. And this is exactly the ductus epididymis in the body of the epididymis. These are besides the clumps of spermatozoa in the lumen of the epididymis. Okay, let's move on. After the ductus epididymis, spermatozoa are transported to the ductus deferens, and during the process of ejaculation, peristaltic contraction of smooth muscle cells, they become they help to transport spermatozoa out. So smooth muscle cells are very important in this process. The mucosa is lined with a pseudostratified columnar epithelium with sparse stereocilia, so they almost disappear out of here, and the adventitium represents the outermost lining. Then also seminal vesicles should be mentioned. They are lined with the pseudostratified columnar epithelium, and that's what they produce, like fructose, prostaglandins, fibrinogen, and that's everything what's needed to nourish spermatozoa and to provide the proper microenvironment when seminal fluid appears inside the female reproductive tract, because it needs to deal with unusual pH level and to provide a spermatozoa with all the nutrients for several hours or even days of incubation there. So they have to survive in the female reproductive system. Then we have to talk about prostate. Prostatic gland is very important, and this is the largest among accessory glands in the male reproductive system, and it is formed around urethra, like the extension of urethra, and its glands are divided into 3 general groups. Actually, one more group is considered, but we concentrate our attention on those 3. And please pay attention. Transitional, central, and peripheral zone. Pay attention that central zone is not in the center, and lots of students confuse because of that. Please be attentive and notice that transitional zone is in the center, and it is represented only it represents only 5% of glands. These are the small glands located in the lamina propria of urethra. Then central zone, it represents 25% of all the glands, and it is located in the submucosa of urethra. Here it is. And the largest peripheral zone, it is located at the periphery, and it includes 70% of all the glands of the prostatic gland. So if we look at the cross section, we'll find out that this prostatic gland, it has a typical appearance like the frozen ice on the window. Sometimes it is compared, and what's unusual with this gland is that lobules are separated not by connective tissue but by smooth muscle cells, because the largest groups of glands, is located in the muscularis of urethra. So here smooth muscle cells separate lobules. That's why with aging, unusual situation happens with prostate, because the hyperplasia of smooth muscle cells results in the compression of urethra, and this is very frequently occurring pathology, and this is the age feature of the prostatic gland. So it is important to comprehend that if usually glands are stroma of the gland is represented by the connective tissue, and usually lobules are separated by the loose connective tissue. Yeah? In case of prostate, in the peripheral group, the largest group of glands is separated into lobules by the smooth muscle cells. So what it produces? Prostatic acid phosphatase, fibrinolysin, it liquefies the seminal fluid. Citric acid helping to create the microenvironment in pH, and prostate-specific antigens. And we highlight especially prostate-specific antigen, just remember this is very important, and prostatic acid phosphatase. Both are used in clinical diagnostics of prostatic cancer. So this is a very widely used analysis, and in case of prostatic cancer is suspected, then prostate-specific antigen is measured. And it is a very relevant sign of the prostatic cancer if it is elevated in the bloodstream. And for prostatic cancer metastasis, prostatic acid phosphatase is measured as like to confirm the same diagnosis. Okay, another feature of the prostate is that with aging, some corpora amylase appears, or the prostatic sand. If you remember, in the epiphysis, in the pineal gland, those brain sand appears, and in the prostate, prostatic sand appears with aging. So, except hyperplasia, also this is what happens with the prostate. So, that's what you have to remember. Okay. And now let's go to the embryonic development, which is very complicated in this case. And first we have to say that male reproductive system is very tightly associated with the urinary system formation. And we have to remember the previous lecture about the pronephros, mesonephros, and metanephros. And exactly mesonephros is used to create some elements of the male reproductive system. So these are the primitive kidney, and in humans it is transformed into male reproductive system. The second feature we have to remember is that before the 4th week, including the 4th week of development, there is no morphological difference between the male and female embryo. So the fate, the die is cast, or the way is chosen based on hormone concentration, based on the gene arrangement at the end of the 4th week. So if there is the Y chromosome, it contains SRY gene, and it stimulates and it orchestrates the male reproductive system development. So let's investigate in details. First of all, what it is written, that intermediate mesoderm forms the urogenital ridges, and those ridges, they give rise to Leydig cells and myoid cells. of the testis. So in the testis, we remember there is the system of the seminiferous tubules. Those seminiferous tubules are formed by invagination of the mesothelium covering those ridges. So it is very important to understand that ridges covering the primitive gonads, mesothelium covering the primitive gonads, they invade inside them and they form Sertoli cells. Then, gonadal ridges themselves, they contain mesenchymal cells giving rise to Leydig cells and myoid cells surrounding the seminiferous tubules. And primordial germ cells, they migrate from the yolk sac of the embryo. So 3 different sources of development. Gonadal ridges with mesenchymal tissue, they give rise to interstitial cells. Then the mesothelium surrounding those ridges and covering the ridges, it invaginates and it forms the system of intratesticular seminiferous tubules, and Sertoli cells arise out of there. And primordial germ cells, they populate gonads by migrating from the external source, from the yolk sac. Then, how testis with intratesticular seminiferous tubules becomes connected to the other elements of the male reproductive system. Mesonephral duct of the primitive kidney, we remember from the previous lecture here, mesonephral duct exists and it is fused with the urogenital sinus, yeah. And excretory ductals, exactly here, they become communicated with the reproductive, and those excretory tubules, they become the efferent ductals in the head of the epididymis. And the mesonephral duct in this region, it is transformed into the ductus epididymis, further into ductus deferens and ejaculatorius ducts, and seminal vesicles, they arise at the extension at the base of those ducts. So it is very important to understand that exactly here we understand how the mesonephros gives rise to the elements of the male reproductive system. And then also we should remember this term at the end of the 26th week, testes, they descend to the scrotum, and this process is regulated by the insulin-like growth factor 3 and testosterone. So those factors are very important, don't forget about that. And if it doesn't occur, and it doesn't occur in preterm born children, for example, or in 1% of the children born in term, in case testes are left in the pelvic cavity, then the temperature is improper for spermatogenesis, and that's what we call cryptorchidism. And cryptorchidism should be treated surgically, and it is successfully done nowadays. So testes are put to the scrotum to allow normal spermatogenesis in future. Okay, in this way we complete the male reproductive system. Next lecture we are going to consider the alternate pathway, how female reproductive system is formed and what's the difference between the two. Okay, thank you for the attention. If you do really like our lectures, please subscribe, and good luck.